Okay, folks, and so last but not least, our final talk in this little series of five is from Andre, and Andre's going to talk to us about some pharmacokinetic. The floor is all yours.
Right. So good afternoon, everyone. My name is Andre Mazzari, and I'm going to talk about my project, Preclinical Pharmacokinetic Studies on Traditional Herbal Medicines, shortly listed for clinical use in Brazil, supervision of Dr. Professor José Maria Pietro. To kick off this presentation, I just want to define the word xenobiotic, which I'm using in my presentation, which is a chemical substance that's foreign to an organism and biological system. It can cause pharmacological responses, toxicity, immunological responses, and also cancers. There are several types of xenobiotics. I can just point out pollutants, food additives, insecticides, and medicines. In case of medicines, when a xenobiotic enters the body, it needs first to cross the membrane, and then there is going to face the first challenge. That's the pig glycoprotein. So the pig glycoprotein can actually pump out back to the lumen, this xenobiotic. And for any reason, if this xenobiotic then is able to cross this first barrier, then it enters the enterohepatic system, and then a series of reactions is going to happen in the liver in order to metabolize this xenobiotic. So first, you have the phase 1 functionalization metabolism that is actually caused by the cytochrome P450 enzymes, and there is a series of reactions like oxidation, hydrolysis, and reduction in order to increase the polarity of this metabolite, and then to be able to be excreted more easily from the body. And then if this metabolite is still not very water soluble, then it forms a secondary metabolite through the conjugation with, for example, glutathione glucuronic acid. All these reactions are meant to increase the polarity of this substance in order to be able to be released in the body, eliminated from the body. So xenobiotics are known to cause pharmacodynamic interactions and pharmacokinetic interactions. And those pharmacokinetic interactions are caused by the modulation of metabolic and transporter proteins. But you wanted to know actually if those effects could be able also causing, if herbal medicines were able also to cause this kind of pharmacokinetic effects. I wanted to use this cover from The Economist, and Brazil's take off. Yeah, Brazil takes off like maybe 10 years, more or less 10 years ago, improving the policies in herbal medicines in the country to guarantee quality and safety of the herbal medicines for the population. And in 2008, the Brazilian Minister of Health issued a list of 71 plant species of interest for clinical use. And then I wanted to find out if those plants could be able to cause any kind of pharmacokinetic effects. And this is actually would be the aim of my PhD. First, I want to review and then to evaluate the pharmacokinetic profile of herbal medicine shortlisted for clinical use in Brazil. But because I didn't want to reinvent the wheel, I needed first to find out which herbal medicines were actually reported to cause any pharmacokinetic effects on phase 1, phase 2 metabolism, pig glycoprotein. And that was the subject of my first review that I published in 2014, The Frontiers in Pharmacology, which I found out that 60% of this previous list have no pharmacokinetic profile. Pharmacokinetic profile of the medicines were not known. The remaining 40% was affecting mostly the CYP4 and glutathione levels, and among that list, none of the Brazilian native species were able to cause, there is no data on effects caused by any of the Brazilian native species. So as you can see here, summarized this table, the herbal medicines which are highlighted are the ones which we found, in fact reported already, and the ones that are not highlighted are the ones that would be the subject of our work. But then, because we are not pirates, we need to ask first authorization for the Brazilian generic heritage in order to have access to those plant species. And then we managed to get authorization to work with these 23 different plant species here. In order to get authorization to work with them, we needed actually to make an interinstitutional agreement between UCL and a Brazilian institution that is the University of Brazil. Brazil. So what we did is literally we acquired these plants from suppliers in Brazil. We sent these plants to the University of Brasilia, where they made the extracts through an infusion of the plant material for 20 minutes. They freeze dry, and then they sent the samples for us to work here in the School of Pharmacy. So here in the School of Pharmacy, we prepare our samples, and we then did our pharmacokinetic studies. First, we wanted to know if those herbal extracts were toxic for our cell models, Hep2 and Kappa2. Then we did the first one, metabolism analysis, is to check if the herbal medicines were able to cause any changes in the gene expression of C3-4. Then we assessed the changes in intracellular glutathione levels, and then the P-glycoprotein activity. First, we have to check then the toxicity of those herbal medicines, and we used actually we found out that those herbal medicines were not toxic. They're very low toxicity. We tested them even at 1 milligram per ml, and they were not toxic at all. So, but the 1 milligram per ml is a very high concentration. It's enormous to work with that. So we had to reduce it, and then we decided to work with a fixed concentration of 100 micrograms per ml for our studies. Then we did the first study on phase 1 metabolism, which to check the expression of C3-4 using relative quantification or PCR quantification. So very briefly, we treated our cells, we did extracted our RNA, we then retranscribed our RNA into cDNA, and then we used beta-catenin to normalize our data. I'm not going to go into details because of the time here for all those results, but I summarized this table here. I can just highlight like a few very interesting results we had, like salic salvo, for example, and passiflora incarnata are herbal medicines widely used here in Europe. As you can see, at this concentration, it was able to increase in 3.25-fold our RNA expression, and passiflora incarnata 1.82. And we want to find out actually if these changes in RNA were caused by the activation of the pregnan receptor, that is a receptor when this receptor is activated by the xenobiotic, it upregulates the proteins responsible for the total detoxification of the xenobiotics, including cytochrome 3-4. And we did a luciferase report assay, and this study is still ongoing. And the only first result that we got is Solano paniculatum, that is a Brazilian native species, was able to modulate activation, was antagonize the activity of the pregnan receptor, which we could correlate with the result that we found here, and the decrease of the C3-4 RNA expression. The other ones are still studied, and we just try to correlate those results. Then we went to the phase 2 metabolism, where we use a very well-known glutathione recycling method. Glutathione is a tripeptide, which is very important in phase 2 metabolism to conjugate with metabolites and then eliminate them as a recapitulated acid. So, and we use this recycling method. Again, there is lots of results here, which are also summarized in this table. As you can see, most of our herbal medicines were able to decrease intracellular glutathione levels. Only one was able to increase. Decreasing glutathione levels could be very dangerous because literally the cell loses one of the biggest defense against free radicals as well, and also decrease the ability of the cell to detoxify the xenobiotics. And again, we wanted to understand if the depletion of intracellular glutathione levels could be happening for lack of cysteine. Cysteine is a precursor of glutathione, and cysteine is more available when we have if you have this glutathione gamma-glutamine transferase enzyme, which will the mechanism is for how it happens. Like we have a GSH glutathione that's pumped out of the cell, and then the gamma-glutamine transferase is going to break down the GSH, releasing cysteine and glycine, which will then be broken down by the peptidase into glycine and cysteine. And then cysteine is going to be brought back to the cell in order to synthesize more GSH. So we're just wondering if this GSH, the lack of the decrease of amount of GSH was happening because of lack of cysteine in the cell. And we did that through an enzymatic assay. Here we have 5 results which we can correlate with our GSH glutathione results as well, because they were all able to decrease glutathione levels, which we could say that the glutathione levels, lowering the glutathione levels, were actually caused by lowering gamma glutamate transferase activity. Those other 3 ones, we still don't know, but we need more studies, mechanistic studies, in order to find out what exactly is going on. The last assay was the rhodamine 1,2,3 uptake assay, in order to find out if the herbal medicines were able to cause, to modulate the P glycoprotein activity. So the rhodamine is a fluorescent dye which penetrates the membrane and is pumped out by the P glycoprotein back to lumen. But once your herbal medicine is able to block the protein activity, so we have an accumulation of this fluorescent dye, and you detect this by fluorescence. And then you have like only 4 herbal extracts were able actually to cause this. You have an increase in fluorescence in rhodamine. That means that you have less rhodamine being pumped out back to the lumen. And this actually is a concern because this is actually can cause interactions. Other drugs that are taken at the same time with the herbal medicine would have their absorption and bioavailability compromised. And then I just wanted to correlate this with what's the biggest point of this project is we need to find out if you're able to predict herb-drug interactions with those results, based on these preclinical results. And none of those herbal medicines shortly listed have been reported by the Brazilian regulatory agency with being able to cause herb-drug interactions. That was another subject of our paper that also has been published in 2014. So the concluded remarks. So the pharmacokinetic data, Brazilian herbal medicines is scarce, and our in vitro results showed that 57% were able to change CYP3A4 expression, 87% glutathione levels, and through the mechanism of effects, pregnenolone receptor and gamma glutamate transferase, 70% were able to modulate P glycoprotein activity. This is the first time that those pharmacokinetic effects are reported, and our data can be used for the elaboration of monographies of those herbal medicines in Brazil that will be eventually can be put into a new version of our Brazilian pharmacopoeia. For future work, we need to do some isolation, identification of compounds responsible for those effects, more studies in the idols, cytochromes, isoforms, and others phase 2 metabolism mechanisms as well, and translation of in vitro to in vivo models, and checking if those effects were causing actually by other mechanisms of action that are not only those ones that we researched. So I have a big thank you list here for all those people who were very special. All those people who were very special during these 4.5 years here in the School of Pharmacy. So thank you everyone for you all contributed with my life here in the School of Pharmacy, and especially for my supervisor, Dr. Mazagasi.
Thank you very much. It's like this to people in the bar on Friday night. The students go on. I'm going to start, it's not very scientific. What I thought was quite worrying, it's very interesting, is 60% of herbal medicines in Brazil, did not have any recognized pharmacokinetic profiles. What would that look like worldwide for all? I mean, is that specific to Brazil, or is that?
No, actually.
Everyone's heard of St. John's wort and things like that?
No, actually those plants actually were being shortly listed by the Ministry of Health.
Yeah.
They are widely used in Brazil. Most of them are actually plants, herbal medicines we use worldwide. Unfortunately, when they create this list, they only they focus more in what we call the exotic plants, where we can find everywhere. But Brazil has the biggest biodiversity in the world, and they were not not actually, this list doesn't focus too much on our native species, which would be more interesting. So those results that we achieved actually were not even reported in papers or not even in the pharmacopoeia. So for the first time, we are checking that herbal medicines are able to cause pharmacokinetic. Pharmacokinetic studies are not a regulatory requirement for herbal medicines, so they are more required for synthetic drugs. And once you disclose those pharmacokinetic effects, you can see that they are, like any other xenobiotic, they are able to cause interactions with conventional drugs. And then we have problems in absorption, we can have problems in metabolism. So the bioavailability of another drug, a conventional drug, can be compromised if you take a herbal medicine at the same time.
Absolutely. Brilliant. Other question?
Just a general one. What was the rationale behind the selection of the plants?
Yeah, the selection actually was made by the Brazilian government when they issued that list. But then, what I did was literally first study all the 71 herbal medicines to make sure that they were not what is known in terms of pharmacokinetic effects of them. Some of them have some studies, few studies. For most of them, there is no preclinical pharmacokinetic data reported. There is nothing. So the rationale when they did this search in Brazil is like they went from region to region in order to find out which are the plant species that are more traditionally used by the population for different conditions. So that's why they reached into this main species, and then they said, well, we need to direct our studies on those plants instead of making our research, doing kind of scattered studies on other herbal medicines that are actually not interested to the country.
Questions? In that case, Andre, thank you very much indeed.